In Chapter 15, we will focus on how microorganisms come in, and the mechanisms that are used amongst all these microorganisms are generally the same. So we're going to talk about how they get in, mechanism of microbial pathogenicity. Mechanism meaning that there are particular steps that these microorganisms will take in order to harm their host. So what are they? Okay. So these are the steps for the mechanism of pathogenicity. We start at the portal of entry, which is number 1. So the microbe has to come in, and this is how they're going to harm their host. And the second one, then they have to get past all the immune system, all the immune defenses that we know and that we've discussed. And number 3, they then damage. So once the damage occurs, this is where you feel it, right? This is when you feel the signs and symptoms associated with the disease, right? When they are causing injury to you, right? So what is that? How do they do it, right? What are the methods used? And then the 4th will be the portal of exit, right? So this is what the microorganism is using in order to harm their host. It is a mechanism, and as I mentioned, generally these microorganisms use the similar methods in order to harm their host. Right? So 1, again, they get in, they cause damage, and then many of these microorganisms will exit. And we'll talk about these strategies and what they are. Okay. For the mechanisms of pathogenicity, the pathogen attaches to the host. All right. So this process, known as adherence, the microorganism and host, they have to stick together. Right? If they do not stick together, they do not recognize each other, and the microorganism will not get in. The foreign invader will not get in. All right, so again, this pathogen, the foreign invader, they have to stick with the host in order to get in. And this recognition between the foreign invader and your host, this then allows the organism to get in and then eventually cause damage to the cells. All right, so the pathogen attaches to the host tissues, and this process is called adherence. This is important for them to get in, and the foreign invader has to stick to particular sites of their host. So let's take, for example, the cold virus. In order for the cold virus to cause its damage and to get in successfully, where does it need to attach? Well, it needs to attach to the mucous membranes of the nasal cavity, right? That's where they'll get in, and that's where they'll cause the most damage and be able to take advantage of the host. All right, and we'll talk about how that will continue. So adherence is very specific. An organism must get to a certain site of the body and attach there. Not anywhere. They have to get to the site where they have particular areas of the whole cell that they can stick to. They can't stick anywhere, and they cannot get in anywhere. All right, adherence to either the mucous membrane or the skin. All right, so this is where they're trying to get in. And once they get past these barriers, remember the mucous membrane and skin, these are barriers, then they can eventually try to keep going past all the 3 lines of the immune system to cause damage. So we're giving examples of bacteria as foreign invaders, and these foreign invaders have adhesins or ligands, and these are on the surface of the adhesins and ligands are on the surface of the foreign invader. All right, so the microbe, remember, we're calling this the foreign invader. In our examples that we give here, it will be bacteria that we're focusing on. All right, so the adhesins, usually these are going to be protein. Right, so mostly made of protein, where ligands, these are mostly going to be made of carbohydrates. And they are found on the surface of the microbe. So surface molecules. All right, for when we're talking about adhesins and ligands. right? So these are surface molecules, also can be called antigens for the microbe. So these are on the surface of the foreign invader. And our foreign invader, remember that we're focusing on bacteria right now, all right? So bacteria, all right, that's what it says on our side. Adherents, bacteria stick to their hosts. But other microorganisms have to stick to certain sites in specific areas in order to get in and cause damage. All right, so the microorganism, they have these molecules and it sticks to the receptors on host cells. So think of the receptors as receiving areas, all right? They're going to receive the foreign invader, so they attach together. So there's different examples of attachment points for our bacterial invaders. For Streptococcus mutans, they have glycocalyx, all right? So for glycocalyx, is this made of a protein or carbohydrate? All right, so we discussed glycocalyx earlier in our class and in our classes as we started the semester. And glycocalyx, remember, sticky. All right, so which one, if you recall, which is it made of? Which organic substance? All right, so the glycocalyx is made of carbohydrate. All right, so Streptococcus mutans has this. In the figure, they're showing you Streptococcus mutans where it's sticking to the teeth. And as I mentioned, the Streptococcus mutans, when they stick in the mouth, we went over this in chapter 10, that these organisms can initiate tooth decay. All right, so they're one of these organisms that start up tooth decay by binding to the tooth. All right, so tooth decay as in our cavities, right? So they're going to eventually start the process of damage to the tooth, all right? So that will be your Streptococcus. Okay, so what about Fimbriae? E. coli, they can use Fimbriae. All right, so let's go over this again just to review. Is your Fimbriae a carbohydrate or a protein? All right, so Fimbriae, these are proteins. Hopefully you got it right. So Escherichia coli, as they show you in the picture in the middle, that these organisms, the Escherichia coli, these organisms are sticking to the bladder cells. And as these, remember the E. coli are rod shaped, as they stick to the bladder cells, they can cause a disease called cystitis. All right, cystitis, itis meaning inflammation. We're talking about of the bladder, cystitis. Okay, and our last example of our bacterial invaders, we have the M protein. And the M protein, as I described in our class, M protein is going to be found in the cell wall of only Streptococcus pyogenes. That's the only organism that we'll discuss in our class that has an M protein in the cell wall. And the M protein is not mycolic acid. This is not mycolic acid. All right, so it's not mycolic acid. But Streptococcus mutans use the M protein, and that can help them to stick attached to their host cells. All right, so just to label this, we didn't label this picture. So this image here, this is showing the phospholipid membrane, and this is your bacterial invader, right? And this will be your host membrane, all right? So they have receptors, so these will be the receptors here in purple. And the receptors will allow them to bind to these antigens, we'll call them antigens for general sake, on the whole cell, right? So those are the antigen, those are the receptors. And they actually have to stick together. Again, this recognition will now allow the foreign invader to be received or taken into the host cells. So remember, one of the most important things is attachment, and attachment is not anywhere, right? For each cell, they have their specific area of attachment, and we are going to separate these attachment sites we're now going to separate the attachment sites into the preferred and parental portal of entry. So what do we mean by the preferred portal of entry and the parental route? Well, the preferred portal of entry is where the foreign invader naturally attaches. They have an attachment site at that point, and they're most likely to get in at that area. So this is the most common way the organism will enter into their host. They attach to a certain area, and commonly they'll attach to the same site all the time, right? So they'll always attach to the same site. So this is the most common site of entry for that particular organism. For the skin, microbes is very hard, remember, to get into the skin. The skin is a tight, waterproof barrier, right? But there are some openings on the skin. So some microbes can use those openings to get in. And the hair follicles and sweat pores provide openings for skin even when it hasn't been damaged, right? When it's intact, it has not been damaged. So the microorganisms can still enter. So some examples I gave, the dermatophytes, which are a set of fungi that cause ringworm. They do this by breaking down the keratin that is in the epidermis. And staphylococcus, these organisms are often known to invade these hair follicles and sweat pores, causing pimples. I'll also add another familiar skin organism. We have, so we'll add 3. All right, the other skin organism begins with a P. Can you remember what that organism is that naturally lives on the skin? So this organism is called Propionibacterium, acne, all right? Acnes, and they can cause acne formation, right? So another skin infection, right? And acne and pimples are different, all right? So caused by different organisms. The initiators are different, so it's different infection. The mucous membrane, as I mentioned, is a common area for organisms to enter because there's lots of nutrients and more water for these organisms to enter. All right, so they like that area. It's better for them to enter that way. Most diseases that we know about are going to be due to organisms that enter through the mucous membrane. Remember that our skin is the opposite. It's not as moist, it is dry, all right? So most areas of the skin is very dry, and most areas of the skin would be dry, and organisms do not survive as well in these dry conditions. So let's focus on our mucous membrane. And the mucous membrane, these organisms, again, they like this area. The mucous membranes that are the mucous membranes of the respiratory tract, these are the most frequently used by microorganisms, and these will be used by organisms such as streptococcus. All right, we also have many cold viruses that will enter, the flu viruses that can enter, organisms that cause bronchitis and sinus infections. They will enter in this way and cause damage. These organisms can be exchanged and transferred to individuals through droplets or dust, right? And some microorganisms using this entry, so I put one here, influenza virus, which is causing the flu, but there's many more as I just mentioned. The digestive tract also has a mucous membrane, and these organisms enter in through food, water, contaminated fingers. Remember, microorganisms are everywhere, right? So they're constantly coming in and having to deal with the digestive tract. But there's so many features of the digestive tract, including the fact that it's highly acidic, that blocks many organisms from causing us to constantly be sick. All right, so some examples we have, giardiasis, all right, and this is caused by giardia. All right, so giardia, this organism we've looked at before, and is this bacteria, protozoa, fungi, or a helminth. Right? So this is a protozoa, right, that is going to be ingested, and when it is ingested, usually it's ingested in the cyst stage, right, in contaminated foods. And we don't know we ingested, but eventually this organism can cause harm. Hepatitis A virus, this is in chapter 13, right, which is one of your DNA viruses. Right, so this is a DNA virus, and this organism can infect, in this case, mostly infecting the liver. Right, hepatitis viruses infect the liver. And the mucous membranes of the genital tract are often used by STDs. And on the last slide, I mentioned E. coli infecting where? Where did it infect to cause cystitis? So it infected the bladder cells, all right? So specifically that area of the mucous membrane. All right, so again, the preferred portal entry, this is where the organism normally enters. They have a site of attachment in that specific region, and this allows them to successfully enter their host cell. So to compare the preferred route to the parenteral route of entry, parenteral route means that when the microorganisms enter, they usually enter when there's a burn, cut, injection, or wound. So there's already some sort of damage to the skin, all right? So the skin, which is naturally a dry area, now is damaged. So when the skin is damaged, the microorganisms are happy because they can get to an area that is deeper in the body, often the mucous membranes, which have a lot of moisture. All right, so I just mentioned and reminded you guys that the mucous membrane is very moist. So they go deeper into the layers of the connective tissue, and sometimes even to the mucous membrane where there's more nutrients, more water for their survival. So they're happy when there's a cut, a burn, injection, some sort of opening where they can jump in, and now they can get the nutrients that they need to survive. Okay, so these organisms, the parenteral route, again, there has to be some sort of damage to the skin, some sort of outside damage. So here are 3 examples. So 3 of them would be here. Right. So the first one, number 1, tetanus, right, caused by Clostridium tetani. Right. So these organisms enter into a cut. Let's say if you puncture yourself on an object, sometimes they give you, as far as tetanus, the example of puncturing your skin with a rusty nail, right? A non-living object where these endospores of Clostridium can survive. Right? So do you remember the characteristics of Clostridium? Is it gram-positive, gram-negative? All right. What phylum is it? What phylum is it in? All right. So Clostridium, yes, it's gram-positive. All right. But what are the other characteristics? You guys, again, you need to go back to chapter 11 for all these characteristics. All right. So you have your rabies virus as number 2. Right. The rhabdovirus enters, an animal bites you. Right. So now there's an opening. The viruses are happy to get into this area where they can grow in those cells and attach there. Okay, so the third one, the example is Plasmodium. Plasmodium causes malaria. This is a protozoa. All right, so this is your protozoa. It will enter. How does it enter? All right, remember the mosquito will bite you. And then in the mosquito saliva, they have the protozoa. So when they bite you, the protozoa goes to you and goes into your blood. All right. So what's the name of the mosquito? Is it male, female? All right. These things you need to know. It's the female mosquito, and her name, Anopheles. All right. That's the genus name, Anopheles. It's a female mosquito. Female. All right. That's the symbol for female. All right. So these are different ways. Tetanus, again, a cut, and then the other 2 are bites. All right. The mosquito bite that we have. Right, so again, the foreign invader must attach. It must attach to a certain site, all right, to get in. And once it attaches to a certain site, the other component that is necessary for the portal of entry is that there has to be a high number of microbes. The more microbes that enter, the greater chance that they will be in surviving in the body. All right, so maybe 1 or 2 microorganisms come in, maybe they're not so successful. But the more that come in, the greater chance they are to work together and survive your immune responses. All right, so we have the number of invading microbes is important for the portal of entry. You have to have a large number of microorganisms for them to tag team together, and then they will get past your immune system. If it's 1 microbe or 2 microbes that try to come across your immune system, chances are your immune system will do its job and get rid of them. All right, so it's very important for these microorganisms, again, in order for them to be successful, they have to attach to the right spot and in high numbers. So the higher the numbers, the greater the success, the more likely they will be able to overcome your immune system. And these microorganisms, keep in mind, foreign invaders, keep in mind, when they are coming in, they're not coming in necessarily, they are not coming in intentionally to damage you, but they're coming into your body to get access to nutrients. And it just happens to be that in the course of coming in for nutrients, they're also damaging you. All right, so really they're not trying to cause harm, but again, you have all these nice nutrients that they want and they need in order to continue their species, survive, and reproduce. All right, so you provide a great environment for that, and that's why they need you. Okay, so our next component that we'll look at is going to be we're going to continue with the second mechanism of pathogenicity, and this is penetration or evasion of host defenses. So remember that these organisms, evasion means escape, all right, the host defenses. And what host defenses are we trying to escape? Mostly those phagocytes, those phagocytes, the white blood cells that are trying to eat our bacteria and other white blood cells, all right? So they're trying to get past the other host defenses. So there's a number of mechanisms that the foreign invader can use, and mostly for escape, these are some of the mechanisms that they're using to escape their phagocytes. And then for penetration, hyaluronidase, and I put collagenase, I put a star in that because collagenase can also be used as an escape mechanism. So we'll look at these different mechanisms as we go go through our next few slides, right? More than a couple here. So it's quite a few slides on penetration and evasion. So I separated them so that way you're a little bit clear. And again, I put a star next to collagenase because it can be used also as an evasion tactic. So the capsule, although it can be used for attachment, it also protects many of the organisms against phagocytosis. And there are several organisms, many of them pathogenic bacteria that are found here, and you can see the corresponding diseases. They use the capsule, and that helps them to be more successful in their invasion. So this phagocyte will try to eat it. So here we see the phagocyte trying to engulf the bacteria, but when there's a capsule around it, the organism either doesn't recognize the capsule because the capsule has familiar substances that the host thinks it belongs to them, or they just cannot digest it. Right? So what happens is it's not successful, and the foreign invader will escape or run away. Right? So it is running away, and more than likely the capsule has substances. That sugar on the capsule is recognized as a host, but they don't recognize it as foreign. So that phagocyte doesn't try to eat it. Right? So again, they get away. Right? So they're getting away. The M protein of Streptococcus pyogenes, this bacteria, is used to resist phagocytosis. So this is another component. So again, the organism will not be able to digest these organisms with the M protein, and they will be more successful in growing in the body. And when the microorganism comes in and they're more successful in growing, remember these microorganisms, especially bacteria, they grow in high numbers, and the larger the numbers, the more likely they are to cause greater damage. So other cell wall components that can protect against phagocytosis, we have mycolic acid. Right, so mycolic acid is the waxy lipid embedded in the cell wall, and this is found in Mycobacterium tuberculosis. And I discussed this particular structure as far as Mycobacterium tuberculosis. They're able to escape, right? We talked about their ability to escape phagocytosis by having this mycolic acid, and they're able to block a particular part of the phagocyte pathway. So what part of the phagocyte pathway are they able to block? So is it attachment? Is it going to be the phagosome? Or the phagolysosome fusion? Right, so which step are they able to block? All right, so we talked about this in the immune system. Well, they're able to block the phagolysosome fusion. So they show you here in this picture, what happens is that the mycobacterium, they come inside, so they're able to be attached and engulfed by the phagocyte. And once they attach and engulf, well, they do get into the phagosome. But what happens is that the lysosome never fuses, all right? So they put an X here, it doesn't attach. So the lysosome, the lysosome, we don't want to confuse it, the lysosome has what? What's in the lysosome that's important? It has digestive enzymes. And those digestive enzymes are able to degrade Mycobacterium tuberculosis if they did not have mycolic acid. But since they have mycolic acid, well, Mycobacterium survives because the lysosome never digests it. So, right, so the Mycobacterium tuberculosis, they're able to block this phagolysosome fusion because they have mycolic acid. And this is what allows them to survive in the phagocyte. As they survive in the phagocyte, what happens? Well, they continue to divide and divide and divide. And once they divide in high numbers, eventually the phagocyte will break and they will be released to go on to infect the lungs in high numbers. So another familiar structure, coagulase. All right, so coagulase. And coagulase is also a factor that protects the microbe against phagocytosis. All right, so with coagulase, you guys have performed this test in the lab and we've talked about it. It will convert fibrinogen to fibrin. All right, so what is fibrinogen? This is going to be a structure in the host cell. So the host is making fibrinogen, and the coagulase, this enzyme, the bacteria, are now taking the host fibrinogen and converting it into fibrin. So again, the fibrinogen, this is going to be found in your whole cells, right? The bacteria now come along, so we'll make a little rod-shaped bacteria, and actually let's put a round shape because we're going to talk about round bacteria. So the bacteria comes with their coagulase, and intentionally they're able to convert it to fibrin. And fibrin is going to be a clotting factor, right? So this is what it is. So coagulase is found in 10% of Staphylococcus. Is this 10% going to include Staphylococcus epidermis or Staphylococcus aureus? All right, well, This will include Staphylococcus aureus, not epidermis. All right, so they are part of the 10%. And coagulase is used to protect them. All right, so here we have it. The fibrinogen is going to be part of the blood. All right, so this is going to be the blood, right? The clotting factors, right? These are part of the foreign blood elements. The bacterium will extract these clotting proteins that are inside of the blood. And what the bacteria will do is that they will surround themselves with this clot. All right, so the clot, they will convert fibrinogen to fibrin, and they will make this clot to protect them. All right, so the bacteria are hiding out. They're hiding out, and this clot, since it's a host substance, the phagocytes do not recognize it. All right, so they don't notice that these bacteria are hiding out. All the phagocyte is going to see is fibrin, and fibrin is a host factor. So they will never know what's inside of it, right? Until the organisms come out. So while the bacteria are inside that clot, they will divide and they will grow and they will grow and they will grow in high numbers. And when they are released from the clot, they will come out in large numbers and they will cause greater damage to the body. Clots can be digested by enzymes called kinases. Kinases in this case are also known as lysins, and these lysins will destroy or lyse the blood clot. And different organisms make different kinases. You have Staphylococcus, which makes staphylokinases, right? So Staphylococcus makes staphylokinases, and Streptococcus makes streptokinases, and these can lyse these blood clots. So these organisms can dissolve the blood clots and release them to areas, right? So they have these abilities to do that. So some organisms can make and break blood clots, others can just only break, but again, this is all part of the bacterial's mechanism to survive inside of their host. So another mechanism that some bacterial cells use in order to protect themselves against phagocytosis is by making an immunoglobulin protease. All right, so before we look at the entire slide, let's look at what do we mean by immunoglobulin and what do we mean by protease. All right, so what is a protease? All right, we talked about proteases in earlier lectures, and this is an enzyme. What does the enzyme do? Yes, well, the enzyme is going to digest proteins. All right, but this protease specifically digests immunoglobulins. And what are we talking about by immunoglobulins? What does that mean? What does that mean? Well, immunoglobulins are antibody proteins. Right? So immunoglobulin protease means that we're looking at a protease that's specific to digesting antibody protein. And who's making the antibodies? Would it be the pathogen, would it be the foreign invader, or would it be your host? Right? Well, these are host proteins. All right, so these organisms are going to be digesting host proteins, and these antibodies are there to protect us. They work with our immune system, right? This is part of our immune system. So the immunoglobulin proteases are used to digest different antibody protein, and the one that we're going to look at is going to be the IgA protease. All right, so this IgA protease is going to be specific for digesting your IgA antibody, right? And there's different types of antibodies. This is just one of them. There are 5 classes of antibodies. So what are the 5 classes of antibodies? We have one of them here, IgA. What else? There's 5. So we have one of them. So there's 4 more. All right, so we have IgG. What else? IgM. IgE. And IgD. All right, so we have 5 of them, 5 different antibodies. That means that these bacteria can make different proteases to destroy the 5 different classes of antibodies set up by our body to protect us. In this case, again, we have the example of the IgA protease that is specifically going to target only one of the antibody proteins, and that's IgA. All right, so the IgA protease is produced by many respiratory pathogens to destroy the IgA antibodies that is found in those areas, right? So the IgA antibodies that are found in those areas will be destroyed, so that allows the organism to survive. All right, so these organisms, these are 3 of them that produce IgA protease. This is Neisseria, gonorrhea, and meningitis, Streptococcus pneumoniae, and Haemophilus influenzae. They all make IgA proteases, and this is what allows them to invade and survive in these areas of the mucous membrane. All right, so again, IgA proteases are going to protect the foreign invader against the immune system of the host, and this will allow them to survive for a longer period of time. Some organisms will use antigenic variation, and antigenic variation, this is another mechanism used by these microorganisms to protect them against phagocytosis. All right, so this will allow the foreign invader to alter its surface proteins. The antigens are on the surface of the foreign invader, and as they're changing, right, these antigens so quickly, the immune system cannot recognize this change, so they cannot see the change. So it's not that they can't see the change, but it takes them a while to recognize the change and then devise a strategy in order to kill the foreign invader. So every time the foreign invader change, well, the phagocyte has to say, look, they changed, so how do we attack them now? Oh, they changed again. Oh, we have to figure out how to attack them again, right? Oh, they changed again. They have to figure out how to attack them when they change again, right? So there's several organisms that can perform this antigenic variation. We have trypanosoma, and trypanosoma is a protozoan which causes African sleeping sickness. The flu is another example, and then the third example that I posted here would be HIV, and they all have the mechanism. Again, once they come into the body, they're there for a little while, so the phagocyte goes, okay, well, we see them, we know who they are, and look, we're going to attack them. But then within a matter of minutes, they change. So are these the same organisms that we saw before? I'm not sure. Well, we need to figure out a new strategy to get whatever these organisms are now, because they don't look like the ones that we saw just a few minutes ago. All right, so as the foreign invader continues to change and change and change, the phagocyte has to develop a new strategy in order to attack them. And it will take them a longer period of time to figure out who this foreign invader really is, right? So that way they can come up with an effective strategy to kill them. All right. So hyaluronidase and collagenase, these are used mostly for penetration, and these allow the microorganism to travel through the deep layers of tissue. Hyaluronidase is going to degrade the epithelial cells. There is a substance in the epithelial cells, it's called hyaluronic acid. And hyaluronic acid, this is going to be sort of the glue that keeps epithelial cells together. And when foreign invaders have hyaluronidase, they're able to break down the epithelial cells because they're breaking down the hyaluronic acid. All right, so here goes the bacterium that's outside the epithelial cell. They'll make the hyaluronidase, All right, and when they make the hyaluronidase, well, they're able to get in a little bit deeper into the tissue, into the tissues. And then some microorganisms, they're able to make collagenase, and collagenase will destroy collagen, right? So collagen, which is in the connective tissues, allows them to get further into the layers of the skin or the mucous membrane. And then at that point, the organism is in an environment where they have access to greater nutrients and water, all right? So the collagenase allows them to get past the collagen, and now they're inside. So we can see now the microorganisms are deep within the tissues. They're happy because they're in an area where there's much more nutrients than outside the epithelial cells, which tend to be dry, and they're exposed to greater damage. So now these microorganisms have gone past the immune system, right? All the immune system defenses, they have gotten past. They've already passed the skin, the mucous membrane, the phagocytes. They escape and they dodge and they dodge, and the antibodies, they got past, and now they can cause damage, all right? So I separate this into direct damage, and then we have toxins, all right? So what is direct damage? Direct damage is just a way of saying general damage by the foreign invader. And the foreign invader is going to cause damage just by being in the host and growing, all right? Taking up all their nutrients, exhausting their resources. The foreign invader is going to cause a lot of damage. So these are, I divided into 4 ways. They can take up all of the host nutrients, exhaust the nutrients in that area, any, let's say, water, nutrients, electrolytes in that area, they're taking it all up for themselves. So that leaves the host cell weak. Some of them will produce toxic waste. And toxic waste is just a matter of them being there, eating and growing, right? They're just secreting all this toxic waste, carbon dioxide, other chemicals into the body that just is not healthy for our cells. All right, number 3, they can disrupt the whole cell function. Some of these foreign invaders, they're able to interfere with protein synthesis. With protein function. Right, so remember, proteins are the workers of the cell, right? Without protein function, then the cells won't work so well. So they're damaging the cells. And some organisms are able to multiply in whole cells and cause the cells to rupture, right? So many parasites, including viruses, right? So many parasites, there are a number of different microbial parasites, but viruses is a good example for that. So again, direct damage is very general, all right? Just the organism being there, growing, taking up nutrients, exhausting the resources in that space, right? Releasing toxic substances as they grow naturally. They are going to be causing damage to the area. So to separate direct damage from other damage caused by toxins. And toxins, these are specific substances made by our bacterial invaders, and they're going to cause damage in a specific way. Right? So let's separate our exotoxins from endotoxins. And the exotoxins are proteins. All right? So they are proteins. Right? And they are produced by the foreign invader, mostly the gram-positive bacteria, but some gram-negative bacteria as well. And they are releasing this as they grow, right? So it's just a part of their growth, and they're releasing these specific proteins that are having harm to the body in a specific way, right? The exotoxins are then released into the surroundings during the log phase. So the log phase means as they grow, right? So they are growing, they are dividing, right? So here goes the cell, right? In this case, they give you the gram-positive cell Clostridium that causes botulinum. and they make a toxin inside the cell. They secrete it outside of the cell, and this toxin causes nerve damage. We also call this Clostridium toxin a neurotoxin. Right? And the neurotoxin is to describe the damage the toxin has to the nerve. Right? It's also called an exotoxin. Right? And we'll give it another name later on. Right? So these are exotoxin. Exo, remember exo, the prefix means what? What does that prefix mean? Exo means to exit, all right? So the toxin is exiting, is going outside of the cell, secreted outside of the cell. So this is what we are looking at, and we give an example of our gram-positive organisms. So to look at some other characteristics of exotoxins, okay, these toxins usually do not produce fever, with the exception of the superantigen. So that's the exception that we'll see. These organisms are usually specific for a particular structure or function in the host. All right, so the toxin has a specific effect. They are usually unstable with heat, right? So they're easy to destroy. And as the temperature in different areas of the body increases, they're not so stable. And as far as immunology, they are neutralized by an antitoxin, all right? So they're neutralized by an antitoxin, all right? So what do we mean by antitoxin? This is an antibody, and the antibody is naturally made by you, made by the host, and that is going to block, neutralize the effect of the toxin. So reduce the effect of the toxin in the host, right? So that way it doesn't have as much damage. All right, so as far as immunology, relationships to antibodies in you will make antibodies to slow or stop the effects of these exotoxins. So what a great feature that our host, our immune system, has already devised a strategy in order to deal with these exotoxins as they come in, as they're in the body. All right, so these are different diseases caused by exotoxins, and we'll get to see many of them later on. Okay, so there's different types of exotoxins. Our first exotoxin, we call it the superantigen. And the superantigen causes an intense immune response. And this immune response is due to the release of cytokines. So the microorganism comes in, will come in, and when the foreign invader is in, then the foreign invader will cause damage by releasing exotoxin 1, which is a superantigen. And this exotoxin 1 will then cause the host to release cytokines. So the cytokine level will go up and up and up, all right? So as the cytokine level goes up, then what we would see is an immune response, all right? So as long as the immune response keeps going and going and going, then we'll see signs such as fever, nausea, vomiting, diarrhea, shock, which is possible, and organ failure or death if the exotoxin isn't dealt with by your immune system or some sort of medication, right? So what is shock? What does this word shock mean? So let's write it over here. Right, so shock is going to be low blood pressure. So low blood pressure, and it cannot be controlled. All right, so you cannot control your blood pressure any longer because these organisms are in your body. All right, so as we get to shock, all right, which is definitely not good, and then organ failure. Of course, many of the people that cannot deal with this toxin in their body, or they're not responding to medication, they can die. So the cytotoxins are bad, all right, as they stay in the body, they're very bad, all right? So we have to eliminate the cytokine. So in order to eliminate the cytokine, well, the body has to eliminate the foreign invader, right? So you have to reduce or get rid of that foreign invader, whether it's the immune cells or some sort of medication, so that way you can prevent the host from dying. The second exotoxin is known as your membrane disrupting toxin. And by membrane disrupting, we mean that it will lyse the whole cell, right? So lyse the whole cell, all right? So the toxin is being made by the bacteria. So again, the foreign invader comes in. The foreign invader is going to make exotoxin number 2. So what happens to the host? The whole cells start to die off. Alright, so this is what will happen. Alright, and these toxins can damage the host lipid layer, the cell membrane. All right, so we have different examples. Leukocidin, they will kill the leukocytes, and by leukocytes we mean white blood cells. All right, an example of organisms that can do this would be Staphylococcus. They can make leukocidin, so they're an example. And hemolysins, these are produced, these hemolysins will kill erythrocytes. Erythrocytes would be what? The red blood cells. Erythrocytes, red blood cells. And streptolysins makes hemolysins. All right, so remember, these can make different hemolysins: alpha, beta, and gamma. All right, so it's a good idea to go back to chapter 10 and review which ones make what hemolysins. All right, which streptococci make what hemolysins. Right. So we have one more exotoxin, and that's exotoxin number 3. All right, so exotoxin number 3, and this exotoxin is also going to cause damage to the whole cell. Specifically, how does it do it? All right, so in order to examine that, we're going to use this model as our reference. And we have 2 cells in the picture. One is our bacterial cell, which is in brown, and these are our host cells that are in yellow. So the bacterial cell is going to make the toxin, all right? They're the ones that are making the toxin. And this toxin is secreted by the bacterial cell. It is your type 3 toxin, or the AB toxin. And the toxin AB has 2 components. The B component is for binding, all right? And this allows the toxin that is released, remember the exotoxins are released, it will then bind to, so here goes the exotoxin is made inside the bacterial cell, it's released outside, and it's going to bind to the whole cell, all right? So here we see it on the outside of the whole cell once it is released. Okay, so now that it is released, this whole cell will then receive it, and it will be engulfed, all right? So they engulf this toxin, it comes inside of the whole cell, and as a result, the A, or active component, is going to inhibit protein synthesis, all right? So it will block protein synthesis inside of the whole cell. So the whole cell is harmed, all right? The whole cell is harmed. Remember, proteins will be the functional component of the cell. So if they are not making proteins properly, right, the cell is not going to be functional. All right, so there's a number of different organisms that produce different exotoxins, and many of these we'll get to see before the last class, all right? So before the end of our semester, all right? So our endotoxins, these are different, and the endotoxins are lipids. They are lipids, Right, so endotoxins are lipids. So let's make a little chart here to compare our exotoxin. Right, so we always want to compare. We want to see what the difference is going to tell us or give us clues on how to answer these questions on our exam. So exotoxin versus endotoxin. All right, so for the endotoxin, these are lipids. All right, so what was the exotoxin? The exotoxin was protein. All right, so that's one difference. All right, so the endotoxins, they are in the LPS layer, lipopolysaccharide layer of the gram-negative bacterial cell. And oftentimes we call this lipid A. All right, so lipid A would be the component that we have. Right, and lipid A is found in the outer membrane. All right, so we're talking about part of the cell wall. All right, so part of the cell wall. So the endotoxin is going to be in the outer membrane. So this means that in this case it is embedded. Right, so it's stuck in the cell wall, and in the case of the exotoxin, so it's in endotoxin embedded, exotoxin released or secreted. All right, so this will be kind of like 1, 2, the second difference. So the endotoxins are going to be liberated when the bacterial cells die, right? And when the bacterial cells divide, this is only a small amount that's liberated in this case, but larger amounts are liberated when the bacterial cell, the gram-negative cell, dies. Right? And the cell wall lyses or breaks apart. So when will the gram-negative cell die? All right, it will either die because the phagocytes are eating it, or maybe they don't have enough nutrients to survive anymore, or maybe you're taking some sort of medication. So these endotoxins will die. So dying will release larger amounts of the lipid A. All right? When they divide, small amounts are being released. So the exotoxins, we also want to add here that they're made by gram-positive bacteria mostly, but some gram-negative. All right, for endotoxins, this will be only gram-negative cells. All right, so only gram-negative cells make endotoxins. All right, so they give you the example of salmonella. All right, so salmonella, this is a gram-negative organism. These are going to be the endotoxins. And these endotoxins will be released. Remember, small amount as the organism is naturally dividing, but larger amounts when they die. Right, so here goes the endotoxins now. They're being released. And these endotoxins, once they're released, then they can have harmful effects on the host cell. So the endotoxins, just to look at some other characteristics, they are likely to cause fever, weakness, aches, and possible shock. So shock means that the blood pressure, remember, is going down. Right? And it cannot be controlled at that point. Right? So this can cause, the endotoxins can cause the blood pressure to go down. Let's see, what else do we have? All right, so they mainly affect cell function, nerves, and the GI tract, as we saw in salmonella. They're much more stable in heat, and with heat, and in regards to immunology, your body cannot neutralize them. So naturally, your body does not make antitoxins to neutralize them. All right, so that means once they're released, they have a greater effect, and they have to get out of the body in some way. There's no antibodies that your body naturally makes in order to protect itself. So lipid A is an endotoxin. I brought this figure back from earlier lectures, so that way you can see where the lipid A is. All right, so the lipid A is in the outer membrane. This is the lipopolysaccharide layer, and they're actually embedded in the outer membrane. So in order to get released, right, they either get released against small quantities when the organism is dividing, but larger quantities when they die. All right, so as we went over, endotoxins are going to be involved in the fever response, and with the fever, in this fever response, what happens is that our phagocyte, so this will be the phagocyte, what will happen, it will eat this gram-negative bacteria, and once it eats the gram-negative bacteria, the endotoxins get released. All right, so this will be the next step, they get released, and what happens is that the host cell starts to make cytokines, so made by the host, right? What cytokine is that? It's one of the interleukins, right? Which one? All right, so this will be interleukin number 1 that is associated with fever and the gram-negative cell. So the interleukins go throughout the cardiovascular system, and eventually we'll get to the hypothalamus, where the hypothalamus will be in control of regulating the temperature. All right, so the hypothalamus starts to release a chemical called prostaglandin, and as the prostaglandin levels stay up, then we would have fever, so the temperature goes up. All right. So how would we stop the fever? Well, we have to get rid of this foreign invader, the one that is causing the problem, right? So the foreign invader is the one that's making the endotoxin. The endotoxin is then causing the host to make cytokines. The cytokines is then going to the hypothalamus and then going to affect this high temperature. So if we get rid of that foreign invader, we get rid of this fever response. All right. So as it mentions here, IL-1 is one of these cytokines that causes fever, but they also mention TNF. All right, so TNF, this is known as tumor necrosis factor. All right, so this is another cytokine that your phagocyte can make, TNF. And the tumor necrosis factor, if it's made by the phagocyte, this can also stimulate fever. All right, so your IL-1, we see that with your gram-negative cells, but tumor necrosis factor can be seen with response to other foreign invaders. Okay, so we've looked through went through the mechanisms of pathogenicity. We started with the microorganism entering. All right, so that's number 1. They get past the host phagocytes and other white blood cells. That will be penetration or evasion of the host defense. They will then damage. We looked at different types of damage. And once they've exhausted their system, once they've exhausted the whole system, or they're under attack by more phagocytes or more immune cells, or even if you're taking medication, these microorganisms want to get out. They want to get out of your body before they are attacked. And they will look for another host in order to continue their survival. So they will exit, right? So they're getting out. So how do they exit? If the organism is in the skin, they will leave usually through the skin. So they will leave through pus or lesion exudate. And then again, their goal is find another host where they can survive, all right? So they'll keep jumping and jumping and jumping to other hosts. So if they enter in through the respiratory tract, chances are they'll leave through the respiratory tract. So they may leave in droplets or sputum. If they enter through the digestive tract, again, chances are they'll leave through the digestive tract. So they'll leave in feces or saliva, and then if they enter through the genital tract, they'll leave either through semen or vaginal secretions or urine, depending again on the organism. They'll leave in different ways. All right, so mechanism of pathogenicity, every organism has a method of how they're going to get into the body, and they're going to use the nutrients for a period of time. And once they feel that they're under attack, either by your immune system or by medication, they're going to go and leave. They're exiting to find another host where they could do the same thing again, right? And continue their survival.